Hi everyone, my name is Taylor Paisley, and for my presentation I will be going over the relationship between clinical and environmental Vibrio cholera in Haiti. So, a little background about cholera as a disease. It is caused by the bacterium Vibrio cholera, and this is a gram-negative bacterium. The disease cholera causes severe acute dehydrating diarrhea, and about worldwide there's 1.4 to 4.3 million cases a year and 28,000 to 142,000 deaths a year. Now, these values are assumed to be underreported since cholera mainly occurs in developing countries, and reporting is usually lower in these countries than the actual number of the disease cases. Now, some of the symptoms of cholera include watery diarrhea, nausea and vomiting, dehydration, and although 80% of people infected with cholera do not show any symptoms of the disease, they are still able to spread the bacterium to other people through fecal contamination. So, transmission of cholera, as I previously stated, it really only affects underdeveloped areas and is transmitted via poor sanitation environments. This includes unsafe drinking water from poor hygiene, and person-to-person transmission of cholera is the most likely way to get cholera. And it mainly occurs in overpopulated areas like Port-au-Prince, which houses most of the Haitian population. And another way is environment to person transmission, because Vibrio cholera can naturally circulate in aquatic environments, and this includes non-toxigenic and toxigenic strains of Vibrio cholera. So the treatment and prevention for cholera has one major treatment is rehydration, and this includes the use of rehydration salts, intravenous fluids, and possibly antibiotics. And this is the main reason why it's no longer endemic in industrialized countries where you have very good access to those types of things. But it's still a big problem in less developed countries such as Haiti and Bangladesh, and Yemen's currently having a cholera outbreak. And one of the main things is we really can't prevent cholera from being introduced. And the main reason for this is because it can circulate in aquatic environments. So cholera, I want to give a little background from a global perspective. So far, there have been 7 pandemics of cholera, the first 6 of the classical biotype, and the 7th pandemic has been the L-Tor biotype. And from this figure, you can see the migration of cholera worldwide throughout time. The 7th pandemic is believed to have started in Indonesia in 1961 and has spread throughout the world, including Haiti. Now, in Haiti, we see a hybrid strain that has been linked to increased virulence in humans, leading to more outbreaks. So why is Haiti such a hot spot for cholera outbreaks? Well, in January 2010, a catastrophic earthquake had hit Haiti, and this did severe damage to their already underdeveloped healthcare infrastructure. Now, the first case of cholera was reported in October 2010 in Haiti, and since then this has led to seasonal epidemics of cholera from this which had been a naive population to cholera. So because of the single source introduction of cholera from Nepal from UN peacekeeping troops, although devastating, it does give us this unique opportunity to study the emergence and spread of cholera throughout the country, and look at the evolution of the genome. Vibrio cholera has many different life cycles, including its environmental aquatic reservoir life cycle and its human host life cycle. Now, Vibrio cholera is found naturally in aquatic reservoirs and is typically non-toxigenic or non-O1 and non-O139 strains that are found in the environment. And these non-toxigenic, non-O1, non-O139 strains typically lack the cholera toxin and its TCP encoding genes. Now, it has been shown that we are able to isolate toxigenic strains in aquatic reservoirs, leading to possibly them evolving in the environment and then able to infect human hosts later on. So Vibrio cholera in the environment has quite a bit of interactions with other organisms and the environment. Vibrio cholera can attach to chitin, and that allows it to become naturally competent, which leads it to many different significant selective pressures, including horizontal gene transfer. And it's also part of the bacterial plankton life cycle in aquatic environments. Um, Also, Vibrio cholera is under predation pressure by protozoa and bacteriophage, and that allows it to become incorporated in the microbial loop. Then low temperatures and nutrient conditions can trigger Vibrio cholera to become in the VPN state, which is the viable but non-cultural state. And this allows it to survive in more harsh conditions. And then Vibrio cholera can also attach to autotrophic organisms such as phytoplankton or microalgae, which can provide a carbon source for Vibrio cholera. It can also attach to chitinous zooplankton and gelatinous egg masses, which allow it to provide nutrients, and these also facilitate potential horizontal gene transfer. Another interesting fact is fish and birds can feed on plankton or mussels, and that might be harboring Vibrio cholera, and this can potentially lead to the bacterium spreading across long distances where it normally could not reach on its own. Here I want to talk about the clinical to environmental transmission hypothesis of Vibrio cholera in relation to the environment. So panel A is the epidemic transmission, in which person to person is the primary mode of spread, and this is how the phylogeny would look if you're sampling all infected individuals in the transmission chain with only person to person transmission occurring. Now, panel B is a mixed transmission model in which the environmental dissemination leads to evolution in aquatic environments, and then this is followed by environment to human transmission and then subsequent human to human transmission. And next to it is a possible phylogeny that could be inferred by sampling all infected individuals in the transmission chain, highlighting both the person to person and environmental to person transmissions. Now, this shows evidence of clustering of clinical isolates with the transfer back to humans. And if we see a tree like this would be consistent with the establishment of aquatic environmental reservoirs for Vibrio cholera in some of the studies that we've done, and then that subsequently leading to human transmission. So the evolutionary dynamics of clinical and environmental Vibrio cholera O run strains, we want to see the ongoing genetic diversification in Vibrio cholera and what is driving the evolutionary changes that are accumulating during transmission within the human populations. And we can do this by using phylogenetic analysis of whole genome sequence data from environmental and clinical strains, which has been shown to be consistent with the establishment of aquatic reservoirs. And of the environmental non-toxigenic Vibrio cholera O1 strains, 2 have appeared to be of the classical biotype, and with molecular calculations this suggests that they have diverged from the main Vibrio cholera O1 lineage in the 16th century. And this could lead to the conclusion that Vibrio cholera O1 strains were introduced in Haiti in the past, but have lost their cholera toxin gene, which has Vibrio cholera cause human illness, but they've been shown to lose this gene over time. So we want to see if the distinct evolutionary and population dynamic patterns of Vibrio cholera from clinical isolates and environmental sources, if they demonstrate independent evolution of Vibrio cholera in the aquatic environment, and if this is causing more outbreaks in humans in Haiti. So here, this is some data I've made with our clinical and environmental isolates. And as you can see, the green dots represent the environmental isolates we've collected, and the purple represents clinical isolates we've collected. And red lines indicate potential environmental to clinical transmission from sample to sample. And you can't really see it here, but they do group by year. And this tree is representing genetic diversity of the samples. So that tells us, if they're grouping by year, that tells us that they are similar in terms of their genetic diversity by year. So they're less genetically diverse the closer they are together. And we do have a lot of evidence of transmission from clinical to environmental and vice versa from this minimum spanning tree, and this tells us that there is potential evidence that Vibrio cholera is evolving in the environment and then leading to that transmission to humans, leading to more human outbreaks. So here's a phylogenetic tree that is scaled in time, and this is showing the environmental and clinical isolates and their evolution throughout time in Haiti. And as you can see, this does support the hypothesis that environmental reservoirs are Vibrio cholera is evolving in environmental reservoirs, leading to human outbreaks. And if this was not the case, the green backbone of the phylogenetic tree would not be green, it would be purple. And the green backbone of the phylogenetic trees suggests that these environmental isolates are evolving in the environment and then leading to the infection of humans later on, which is very interesting, and the tree does support that hypothesis. So with this figure, we're looking at the clinical environmental effective population size. But this figure, instead of really representing the effective population size, it more represents the relative genetic diversity of Vibrio cholera. And this plot is a method for estimating past population dynamics through time from a sample of molecular sequences without dependence on a pre-specified parametric model of demographic history. And the time is measured in units of mutation per site. But so as you see a peak, that more represents more genetic diversity in the pathogen, in this case Vibrio cholera. So if it goes down, there's less genetic diversity. But as you can see here, we see spikes where environmental isolates have evolved and then potentially have led to more genetic diversity in the pathogen, which is also led to more outbreaks in humans, potentially. And this deserves a little more research into, but with our data so far, seems to be how we do see the evolution of Vibrio cholera in environmental reservoirs. So everyone, thank you for listening. That's the end of my presentation. If you have any questions, feel free to comment and I'd be happy to answer. Thank you very much.